Hi, everybody. It's Mark Swain. Welcome to the webcast today. It's the afternoon now of the 12th of November. We're here with our old friends Algernon, CEO Chris Moreau, and Vice President Chris Bryan, and a special guest today, Dr. Rick Strassman, a famed neuropsychiatrist, author on the subject of dimethyltryptamine, and expert on that and hallucinogenics in general. We're really looking forward to hearing from him. It'll be a panoply of topics today. Algernon has had a busy year, to say the least. Number of irons in the fire there. In fact, I started to subtitle the talk that today, Irons in the Fire. There are so many things going on with ifenprotal, with the DMT program, and I think Mr. Moreau will update us on that to a degree today, to the extent he can. The company is entertaining the idea of listing to NASDAQ, so we may have some constraints on how free with the information he can be on that subject, but suffice to say, the company is busy, productive, and I don't think Chris is not tired when he lies down at night to sleep. So he's we're earning his keep. Dr. Strassman, it's really a pleasure to have you here, have a person of your stature, your caliber, and we look forward to a really lively discussion there. We are a bit talk show-like, a bit conversational, and if we jut in now and again with a question, it's just kind of our way of doing things. We're not trying to be rude, but let's get rolling. And Chris Moreau, you want to open, or how should we do this?
I just want to thank Rick for joining the call today. I think in the early days there was a chance for us to chat with Joe Rogan, who has an interest in DMT and so on, and that didn't work out for timing. So I'm glad it worked out today with you, Doc. But glad to have an update. I just think at a high level, we're progressing quite quickly with our DMT stroke program. Chris Bryan and I were in London, UK last week. Sort of interesting to get back on the road. I've taken a few trips in the last few months, but the first trip overseas, and it was actually well organized, seamless in terms of PCR tests and getting into London. But more importantly, we went to meet with Hammersmith Medicines, where we are planning our phase 1 study for DMT, which is really safety and dosage. And Chris can chat a bit about the details. We are hopefully in the next week or two, we'll be able to announce the completion of the cGMP synthesis of our DMT supply. Of course, you can get research grade DMT to do preclinical, but for human clinical trials, you need cGMP material. We got very lucky with Dalton Manufacturing in Canada. There's been so much interest and pressure for licenses to manufacture psychedelic drugs that Health Canada apparently really, they were inundated with applications, and we got our application in, and we've had a very smooth process with Dalton. So we expect that material to be ready shortly. And we've also have a meeting next week with the MHRA of the UK to talk to them about our phase 1 study for DMT. So bottom line, we're looking forward to a phase 2 study, hopefully as early as July or August next year. We don't think we're going to have any issues with our phase 1. There have been a number of phase 1 studies, including the studies Dr. Strassman did years ago. So I think it's very predictable that Algernon will be running a phase 2 study for acute stroke in Q3 of next year, and that's a very exciting proposition.
I agree, Chris. That's great to hear. Dr. Strassman, I think your name has considerable marquee value because we've got the attendees piling on now. So, Chris Bryan, Vice President, do you have anything shall we hear from you briefly? Anything to add?
I mean, I could give you more details on the meetings, but I don't know how exciting that would be for your viewers. Other than say we were pleased with the facilities at Hammersmith. This is the first time we'd had the chance to tour there, and I'm confident that we're in the right place and they've got the right people to seamlessly execute our study. So it's just a question now of various paperwork winding its way through the various bodies, and we expect to be underway ideally January of next year. We're going to push for that. And that's our target.
Right. I've genuinely enjoyed working with you, Chris, this year. You are wicked smart, boy. We've had some lively correspondence discussing some things about drug development, and I'm impressed, and I'm glad you're with Algernon. I really mean that.
I appreciate the kind words, Doc, and hopefully now that the borders opened up again, you and I will have a chance to sit down in a less formal setting and talk about various topics.
Hope so, indeed. That'd be great.
Doc, I just want to jump in on one other little update. We have been working on a preclinical study with Charles River in Finland. They have a license to work with DMT. It's a real challenge to find access to the drug, even research grade, to be able to do the research. The restrictions, as Rick knows, and also David Nutt, when you have this classification of psychedelics, yes, it does restrict the general public, but it really restricts research and science to get access to the compounds to be able to do important work. So we've been focused on trying to understand three things. One is what's the best concentration of DMT for a stroke study, and we plan to deliver it IV. And second is what's the duration of that treatment? There seems to be an initiation phase, and then as the hours progress, there seems to be a progression of neuroplasticity and going on. And then third, what's the right timing? Is DMT protective? Does it help during the insult? Does it help with reperfusion injury? Does it help a few hours after? We would like to get some more clarification in our phase 1 study, whether DMT increases blood pressure and heart rate, which would mean it may not be ideal for a hemorrhagic stroke patient. But in Dr. Strassman's work, it seemed not to have in the lower doses. And just to remind everybody, we are working with sub-hallucinogenic doses, sub-psychedelic doses. We don't want to, a number of stroke drugs have failed because people actually have been jumping out of beds. They are hallucinating. Even in a clinical setting, they're not set up to be also monitoring for people going on a trip. And you've already had an injury. So what we're excited about is that in our own independent study, we saw the most effective dose at around 30 nanomolars. And if you translate that into a human dose, it's well under the 0.1, 0.2 milligrams per kilogram that Dr. Strassman identified as sub-psychedelic in his studies.
Okay. Chris, do you want to introduce our main guest today, or how shall we do that?
You go ahead, Doc. You're much more prolific than I am with your
Dr. Strassman, I will confess not having read your book yet. It's on the night table to read, but we are indeed gratified, flattered to have you here today. We'd love to have you back anytime. And I think you'll find this a very receptive audience, probably with a lot of questions. And we'd really love to hear your take on what Algernon is doing on the space in general, given the profusion of companies now dabbling in psychedelics, and where you think this is taking all of us.
Well, I can give a little introduction to my work. I mean, in college, I was interested in starting to kind of nail down the biological bases of spiritual experience. I was a product of the 60s and on the West Coast. There was meditation, there were psychedelics. And, to the extent that there was an overlap between the two sets of experiences, I believe there was some biological common denominator too. So I began my clinical research career studying melatonin. There was some evidence it was psychedelic back in the day. That didn't turn out to be the case, and I pivoted to DMT. And our study was the first new U.S. clinical research with psychedelics in a generation. We ran the study from, it was a 2-year process of getting permission from FDA and the DEA. And in the meantime, I got some private funding and some funding from NIH as well. So the study ran from 1990 to 1995. We studied almost 5 dozen normal volunteers, gave 400 doses of various strengths of DMT and some dose response work in a group of normal volunteers with previous experience taking psychedelics. And we generated a slew of biological and psychological data. So some of that data is the basis of Algernon's work and the Hungarians' work. Looking at sub-psychedelic doses of DMT as either neuroprotective, stimulating neurogenesis, stimulating neuroplasticity. And it's been quite promising so far. The basic science data is there, and it's being fine-tuned. What kind of doses are effective, what kind of duration exposure. There's been an explosion of interest in the psychedelic space for a number of reasons and for a number of indications. So I think it's a good thing. The more treatments are out there for conditions which are otherwise difficult to treat, including stroke, TBI, dementing diseases, all those things. Psychedelics, may provide a unique category of compounds, and especially if you can minimize the so-called adverse effects, which translate into tripping psychedelic effects in people that you don't want to cause those kinds of experiences. So if you can have the neuroprotection, the neurogenesis, without the psychedelic effects, I think that'll be the sweet spot.
Do you think the current resurgence and interest in psychedelics, does it represent meme propagation, or does it represent neuroscience having come to a point of critical mass where we've matured in our take on it, we're ready to address the frontiers that these things provide in a way that we weren't, say, 20 years ago? I mean, why is it happening now and not yesteryear or 5 years from now? What do you think?
Well, I think that the first generation of the first wave of clinical research was a bit naive, and I think we've learned a lot in the meantime. One of the reasons I think too is the stigma of psychedelics isn't anywhere near as great as it used to be. Once these compounds were scheduled, they basically went underground. And a lot of urban wisdom kind of built up around them in order to minimize adverse effects. Smaller doses, better preparation, knowing your source, being able to manage bad trips more readily. So the negative press that was associated with psychedelics back in the day began receding from memory. I think the advances in serotonin pharmacology, which occurred in the interim between 1970 and 1990, were dependent on the psychedelics, especially LSD. So that people started to appreciate the information that could be garnered from studying psychedelics in the world of basic science. And, our study was quite, meat and potatoes. There was no flourish at all behind it. It was, dose response, neuroendocrine effects, cardiovascular effects, develop a new rating scale, which was free of ideology. So we established a model for studying psychedelics that was rigorous, could be funded, could be approved. And it, started a small flame of clinical research. There was, some OCD work in Arizona, some Ibogaine, work in Florida, MDMA work at UCLA. But things really took off, once Hopkins, started publishing their data. And Michael Pollan's book, So in a way, it's a good thing, but I think at the same time, we need to be level-headed and, not make, the same mistakes, which were made, way back then. You don't want to glorify potential benefits and you don't want to minimize, potential risks. So I think if we can, keep, science in the forefront, we'll be, much less likely to make the same mistakes. Also, a lot of people that, took psychedelics are now in power in the government and, the regulatory agencies and the media, so they'll be more kindly disposed, to the field. Plus there's money to be made. There's a lot of mental illness out there, especially with the pandemic. There's a lot of brain injury, from combat and from sports. So, it's in a way the perfect storm, for a resurgence of, clinical psychedelic work.
In your view, having seen what you've seen over a career, is there any evidence to say that delving into lapsing into the hallucinatory experience with a psychedelic agent, does it represent neural circuitry being floorboarded? Does it represent a toxic manifestation, or is that just a gross oversimplification?
Well, the toxicity of the classical, psychedelics is, pretty much, zero. You can take a pound of, well, I wouldn't say a pound of LSD, but nobody has ever died of an overdose of LSD, pharmacologically.
Interesting. Okay. Didn't know that.
DMT is not neurotoxic. Psilocybin is not neurotoxic. Mescaline is not neurotoxic. So they're safe drugs, and the psychedelic response is activation of the relevant circuits and receptors in the brain. Whether the subjective effect is necessary, for the benefit is an open question. And when you're speaking about, neuroregenerative properties, the psychedelic effects, the subjective experience, may turn out not to be necessary.
We know someone well here, and I probably should have invited him today. I won't reveal the name because I'm not sure he would be okay with that, but was around at a time before the FDA had imposed, rulings on the disposition and availability of the psychedelics, and trafficked in them to a degree himself. And he described very eloquently being with a group of other teenagers on a beach and seeing 3 young women who were sort of engaging in girl talk, kind of cackling, and they all transmogrified into chickens right in front of his eyes. Certain symbolism there, and he really speaks of their effects in rather fond terms. Not certainly, you know who he is. Certainly, no question that it's not left him harmed. So yeah, it's interesting to hear you outright declare that they're not neurotoxins. So
no, the classical psychedelics are not neurotoxic.
Okay.
Well, and in fact, they seem to be
neuroprotective,
regenerative.
Yeah.
Doc, just on to circle back a bit on the market acceptance and regulatory, we took a bit of an unusual step. We were planning, I think, almost from day one to do the phase 1 in the UK. We announced our DMT stroke program on February the 1st. March 1st, we announced we had retained Hammersmith Medicines. We had brought on board Dr. Strassman, Dr. David Nutt. Dr. Nutt had helped us connect with Hammersmith. Hammersmith had done a DMT phase 1 for small pharma for their own formulation for depression. But we also filed a pre-IND with the US FDA. I mean, ultimately, that's the biggest market in the world. You want to know what they're thinking. At some point, our plan would be to, if we're going off to the UK to do the research, to come back with the data and say, okay, can we move, do we do our phase 2 in the UK and then come into the US for a phase 3 if we're successful? The pre-IND feedback was a bit surprising in terms of some of the limitations, and we shared them with Rick. it's a bit surprising, even because of the current climate. There seem to be a number of academic institutions doing research in the US with psychedelics. Maybe it's different for a sponsor doing the research directly, but it was, some of the concerns, how they addressed what their concerns were. It showed a certain attitude still that
Parochialism.
What's that?
Parochialism, is that what you're saying? Maybe?
Standard, and concerns, sort of ignoring perhaps the fact that this isn't the first in man trial.
Right.
Expect sort of comments on toxicity, and I'm not criticizing the FDA here. I'm just sharing they had concerns that I think showed consistent with the fact that this is a scheduled drug and so on. But many scheduled drugs, its current schedule means there's no therapeutic use. So if you can establish a therapeutic use, then there's a process for approval. And secondly, just one other comment. I had organized a series of small information spots on BNN for last week that we're going to run for the week. There was basically saying Algernon is working with the psychedelic drug, and BNN said, no, we're not letting you use the word.
BNN is, sorry, I'm an American.
The business network, it's run by Bloomberg, so it's
okay.
business CNN.
Okay, but it was a Canadian thing. All right, never mind.
And I also think the Financial Post was sort of restricting use of the word psychedelic. And
what did they accomplish by doing that, Chris? I don't understand.
Concern of, well, look, you follow the money, right? Does that mean they're going to get pushback? I mean, it's all basically fear from the fear that these drugs are going to be approved and we're going to have issues, anarchy, people behaving, the way that they painted how psychedelic drugs, and look at, they're powerful compounds. They have to be understood, but you also want to have access to a pure API of the drug and all of the things that come with a managed program. But probably just a fear of sponsors maybe leaving them, or advertising sponsors not wanting to be associated. It's too early, so it could be a while. I mean, we're breaking ice, I think, and we're glad to be a part of a group of companies that are moving forward. But it's not. It's going to be, we've got some heavy lifting to do.
It's a bizarre thing.
You as a company are not advocating Black Sabbath albums and anarchy and pregnant widows, right?
I mean, we're not making a case, any comment about the psychedelic elements. We're actually focused on the sub-psychedelic. That's just
Sure.
our clinical focus.
Well taken. Touche.
There's this strange, kind of almost like this reefer madness culture is still, in hold almost in certain areas, which, almost a century has passed since that came out. You would think that we'd be a little more advanced, but, there was some very interesting psilocybin data that came out very recently that for depression, and, there's very real uses for these, assuming that we, go through a controlled trial setup like we would for any other drug, but there's still this just culture, that parochialism that's just against this entire thing. It's very strange.
Yeah.
I do wonder if the recent process, which has happened in Canada and now is happening in the United States with the drive to legalize cannabis and marijuana, is affecting the attitudes of regulators. Of course, it was prohibited for a long time. It's now come to the market, and people are making all sorts of claims about the various potential therapeutic effects of both cannabis and CBD oil, all of which very much are in advance of any clinical program. And I wonder if the authorities might see some parallels between a segment of the community, which is not Algernon, but who would seek decriminalization and recreational use of these drugs, and they're pushing back against that sort of pathway opening up.
Good point. Okay.
Yeah, that could be. I'm a little bit skeptical of the decriminalization and legalization movement. I think it's going to inevitably increase reports of adverse effects. and that's going to gain or garner media attention. It's a bit it's a bit surprising FDA is so skeptical, because they did approve a study at Yale. They're giving IV DMT in a psychedelic dose, for depressives. So for them to ask you to go back to to the drawing board, both in the setting of my work, which established safety, and the Yale work, which is ongoing, at the present time.
Yeah.
Chris, for your actual trials, will you be able to use the work that Rick has kind of put together for like your own safety package?
Well, it's whatever's part of the public record.
Sure.
We put together in an investigational brochure, and then we have supporting evidence and documentation.
Mm-hmm.
So our first step is to have this scientific discussion with the MHRA. It's akin to a pre-IND meeting with the US FDA.
Okay.
We get their feedback, then we will formally apply for the phase 1 approval of that study, and that should happen quite quickly. But yes, all of the documentation, all of the research and data that we have always goes into the background file and background information, and helps support your hypothesis or a claim that you're making that, as an example, we don't need to do genotox, we don't have to do basic tox, and that we can move it quite quickly into a human study. And that was what interested us about DMT, because Algernon, after all, has been focused on repurposing drugs. We're sort of, DMT has never been approved, but we're to some extent repurposing it from its psychedelic secular activities
to neuroregenerative,
to a clinical focus on helping to treat stroke patients, from what all the the work Olsen did and Nardi and so on, and Strassman. And this is all but yes, we do aggregate it all, and it goes into the package, and the more details we can get, the better. And there's very little. And then of course, having Dr. Strassman on our advisory board also helps, because
sure, yeah,
we're also he's a resource to us, and also we let the regulatory agency, they also know that we have some of the top guys in the world who have worked with psychedelic drugs, and specifically DMT, that are available, helping us to advance the science.
Well, it's a bit ironic, that FDA is requesting a lot of, preliminary data. When we applied, to do, my study, I just adduced the previous human research from Hungary, from the US, and said it's been given safely to humans in peer-reviewed scientific papers, and I didn't need to produce animal data. They were just mostly interested in the synthesis and the purity and the precursors, and the identification of the compound, and the quality control afterwards. But they weren't interested, or they didn't ask for preclinical toxicology at all.
It's bizarre. I mean, that's decades ago, and that's almost a more conducive atmosphere then than now, which you would expect the opposite.
Yeah, I mean, I got everything going in the middle of the war on drugs, the first George Bush, and that kind of environment.
Wow.
Not to kind of bog down the conversation, but given the fact that Biden's appointed a full-time FDA commissioner, do you think that maybe there'd be kind of a little more leniency, or do you think it's going to be just more of the same?
Well, do you know who that is?
Robert.
Yes, it's the more things change, the more they stay the same. It's Robert Califf again,
who I know personally, in fact. Yeah.
Well, so what is his approach to psychedelics? Because some people at FDA are quite open-minded, and some people are not.
I think Dr. Strassman, he will be non-doctrinaire. He's very by the book. T's must be crossed, I's must be dotted. He's very rigorous like that. I don't think he comes in with a preconceived opinion about psychedelics, though. He's a cardiologist, sort of a cardiology epidemiologist, and really was kind of a person who pioneered the idea of the corporate clinical, corporate academic collaboration to produce big phase 3 studies and advance careers academically, get tenure. And that was kind of the mode that he brought along. And I think he's capable of being an excellent commissioner. He really was too short-lived in the job previously to, I think, flesh out much of an agenda. I'm not going to say anything bad about him because he's been very decent to me over time, and I think really is viewed in a paternal way at Duke, where he's from. Yeah, so
Interesting. Well, the acting commissioner was Janet Woodcock, right?
Yes.
yeah,
Yeah, so she oversaw the psilocybin work, the MDMA work that's been going on the last 20 years. So it'll be interesting to see.
Dr. Strassman, when you saw the Algernon nerve regrowth data, the sproutings out in those pictures that were so dramatic, what was your reaction to that? Given that, hardcore, there it is, proof positive of an effect. Maybe you'd seen that effect before in other systems, I don't know, but
well, one question which I'm occasionally asked is, is neuroplasticity and neurogenesis a good thing? Could these nerve cells be popping up in the wrong place, and are the connections for
certainly be downsides to it, yes.
For ill, but if you make the connection between the in vitro data with the functional recovery data, the reduced stroke size data, it isn't proof of causality, but there's certainly a strong correlation between the neuroplastic genesis effects and functional effects.
Right. I mean, were you surprised by the dose that they invoked having the effect that they saw?
Well, not really, because the Hungarian data indicates you could give subpsychedelic doses. The David Olsen data is subpsychedelic doses. So it's all pretty consistent.
Yeah. Okay. Jonathan, what do you? Oh, Chris, go ahead. Yeah.
I just wanted to say that the study at Charles River, the preclinical work, or in vitro, was an expensive study, but we wanted to do it because we wanted our own data as well.
What made it particularly expensive, Chris?
Probably a number of things. There are certain licenses. There are very few companies in the world that can handle DMT that have the license to do it.
Okay.
The cortical neurons and all of there's three elements to the research we were doing. First was concentration. Second, we did a duration study where we exposed the neuron. The first exposure was 1 hour. That's where we confirmed the 30 nanomolars. Then we did 3, 6, 12, 24. And then the third study we've been working on is trying to confirm mechanism of action. Sigma 1 and blockade of Sigma 1. And so there was a larger study, but we probably could have gone to the MHRA just with the data in the work that had already been done. But really, we also wanted to be able to confirm, and we wanted to direct our own phase 1. It's nice to have the work that's been done before. It's also nice to validate the research, but
right.
That's why we undertook the preclinical work ourselves. But just to add to Rick's point, while Olsen initially exposed cortical neurons to LSD, DMT, amphetamine, it was really the Nordic study where they had the rat occlusion stroke model, where they then exposed the rat to DMT very shortly after the insult. And then after 30 days, there was this almost full recovery of motor function, reduction of infarct volume. So, but it's interesting, the question, because how can you answer every single question? Sometimes you can, sometimes you can't. So somebody could say, yes, it promotes neurogenesis or neuroplasticity, but does it help?
Right.
I mean, is it happening in the wrong areas of the brain? I mean, wow, it just so many different angles. But from the at least the animal study, it seems to correlate that the effect of DMT had a clinical outcome, positive outcome.
I mean,
Is it
Chris,
having
that's
effect
very
on?
striking. I mean, it's really that's a you said that casually, but that's really a sweeping statement of sweeping significance. Really, could be quite ramifying, right?
Sorry for using the word clinical outcome with a rat, but you're getting the the other important point I think that we can and I've just, by the way, I get about 5 to 6 emails every day, and one of our board members is a medical doctor, and my replies always include him on the CC of people saying, I've had a stroke, I've read about DMT, could it work, I'm taking care of my husband, there's nothing for us, could we please be a part of your study, we'll fly anywhere in the world. And it's really heartbreaking to read, of course. This is such a needed area, and I've said on a previous interview, an analyst asked me why in the hell we would be working in stroke since hundreds of drugs have failed, and I said to him, it's because hundreds of drugs have failed,
Right.
and we can very quickly be in a phase 2 study in 6 months, 7 months. We
But by the
expect.
same token, hundreds of drugs have not succeeded in these early stage trials. Am I right?
Yeah, I don't have a number for that, but lots of, that 90% of drugs never make it out of preclinical.
Right.
They just don't. It's a horrendous, it's very expensive, it's high risk. So from a drug development model, if you can take a compound like DMT, where you already know you're not concerned about a safety or toxicity signal, and you can move it into human trials quickly. Mark Williams said to me many times, Chris, we'd be sitting around and say, maybe we should do another animal study. And he said, let's get it into man. That nothing will replace the complexity of a human being and trying to get, as long as you know it's safe, you can quickly move it into a human being, because animal studies are limited.
Yeah.
Chris, obviously, your comparator here in the stroke field is just going to be TPA, which is pretty much a fairly crap drug, no offense. So, I mean, you've got a pretty easy bar to clear once, you get into kind of, into the clinic. What does, I guess, next 6 to 8 months kind of look like for DMT, phase 1 and then possibly phase 2?
Yeah, so phase 1. So when Chris, Brian, and I were in UK last week in London, we met with 2 top stroke experts introduced to us through Hammersmith, and we had a better understanding that in London there are 8 hyper stroke centers, so that if you're suspected of a stroke, you're directed to one of these centers. May even be larger than London. Takes about 15 minutes to have access to ambulance, about 15 minutes to get into the stroke center. We started to talk about when we could actually deliver DMT in an acute study, because right now, until you have a CT scan, they need to know if you have a hemorrhagic or an ischemic stroke. About 15% of patients are hemorrhagic, which is a bleed.
Yeah.
85% are ischemic, that's a blockage. And they can't give you a TPA, a clot buster, or blood thinners if case you have a hemorrhagic stroke. It would be very damaging. So we were talking about how quickly might we position the phase 2 trial for DMT before the CT scan, where it doesn't matter if you've had a hemorrhagic stroke or ischemic. DMT, as long as it's not increasing blood pressure,
Right.
Increasing heart rate. What we'd like to find out is, how soon do we have to give DMT? Does it matter if it's within 30 minutes?
Yeah, your window of opportunity here. Yeah.
We have to work with standard of care. We have to make sure that DMT is not negatively impacting blood thinners or TPA as a modality. So we have an arm of that study. Can we give it a few hours after? So after the CT scan, now it's a larger study. You've got more arms. So you've got the ischemic, it's binary, ischemic, hemorrhagic. And so we were talking a lot about, with these experts, where we could position it. The second part of the study that we'd like to, or a different study, is, can we help reduce the deficit after sort of the stroke event has occurred and the first in line treatments, TPA, trying to stop the bleed. Now you're into a therapy, and in England they keep you for 1 or 2 or 3 weeks. we were sort of thinking just about CIMT, that's constrained induced movement therapy, where if you have a affected limb, they will, for a few hours a day, tie off your healthy limb and force you to use the limb that's been affected by the stroke.
Right.
And we thought, well, maybe we could give the patient an IV dose of DMT ahead of that, and every 2 or 3 days, give them another IV dose, and then see how well patients that are getting DMT do over control or placebo. But in talking to some of the stroke experts in London, they were suggesting, look, it's ideal, it's a clinical setting. We could give DMT to the patient. They may have confusion. They may have lost some of their sight. They may have other motor function or cognitive deficits. Maybe DMT could help them as well. Let's not limit it to CIMT therapy. So, we'll be working hard in the next few months to sort of figure out, once our phase 1's done, what's the best phase 2 study in the acute and in the post treatment or post injury, the therapeutic phase, and then try to figure out what's the best. Hopefully we can have access to enough capital to run both studies.
Yeah, that's obviously going to be another issue. I'm sure we'll have some people asking questions about how ubiquitous is mechanical thrombectomy in England. We know of another company that ran quite afoul of expectations because they forgot to take out that question around stroke therapy.
I don't have I don't have numbers for you, but you always have to take into account standard of care.
Yeah.
When you're introducing your new potential therapeutic, and you can do a lot of work ahead of time, and so that you don't run afoul of that. Another issue too is consent. And we had a better understanding of what percentage of patients who are ischemic are conscious that could actually consent. At one point we were thinking you could even deliver DMT in the ambulance, but it's different in every country. In the UK, they had a big fight to allow ambulance drivers to deliver aspirin. So we don't want to go down that path. So by working with these hyperacute centers for stroke, then we don't have to worry about that. So there's a lot of complexities to it, but we're thinking it all through.
Jonathan, that intervention, of course, requires well-trained vascular radiologists. I don't know the UK has that kind of vascular radiology workforce. I don't know. It'd be interesting to look.
That's kind of my question, considering that, they're talking about the largest centers there in England or London, specifically, they probably would. But if you get more into, kind of outside of, that general dense population, that may be less of a concern.
One of the things we learned from the DMT work in New Mexico is that subpsychedelic doses don't raise blood pressure or heart rate any more than placebo does. So even in the case of hemorrhagic stroke, I think the risk of increasing the size of the infarct wouldn't be as great as otherwise might be the case if these subpsychedelic doses were hypertensive.
Good news.
Dr. Strassman, while you're here, I wondered if I might, prevail on you to kind of mine your brain and your experience a little bit about two issues that have occurred to me, and others feel free to chime in or joining as we talk about this. But you alluded early on to the psychedelic experience as having, aspects in common with religious experience, and we know that there's the real world and there's the molecular milieu, the neurochemical milieu with which it's experienced, which is modified by the real world, and vice versa. The neurochemical milieu acts like a kind of chemical homunculus, if you will, for the external experience. There's a barn door that swings in both directions. We know that if you are a chronically depressed patient whose face is Botox so that you can't frown anymore, you experience abatement of the symptoms of depression, that sort of thing. So if psychedelic drug administration can be experienced as a quasi religious thing by the patient, my question is, for the patient who's experiencing the genuinely religious event, is there a definable change in neurochemical milieu that has been explored? And if so, what does that tell us about religious experience?
Well, there's quite a bit of interest in the so-called mystical experience out there in the psychedelic research world. But I think it's more accurate to call it mystico-mimetic. It resembles, features phenomenologically of the bona fide mystical experience, which occurs in, tradition. And in those traditions, the mystical experience confirms the tradition, rather than being the goal of the tradition. So facsimiles are very powerful. I've got a facsimile Van Eyck's Annunciation in the corner of my room, and it's a beautiful, inspiring print.
Right.
It isn't the original, but still it has got an effect. So there clearly are correlations between scores on mystical experience questionnaires and clinical outcome. So the functional neuroanatomy, neurocircuitry is being worked out. I'm not that current or conversant with brain imaging, default mode, network, kinds of data, but there are correlations. There are correspondences between any subjective experience that's brought on by psychedelics and the neuroimaging data.
Oh.
Well, you keep in mind too the so-called explanatory gap. We can understand the pharmacology, the physiology in great detail, but how that translates into subjective experience is still unknown.
The reason I say it is because certainly, even as sophisticated as we think we are now, I don't know that I can think of a single person who really asked personally about it would deny having had mystical experiences. They seem to be universal. Would you agree with that?
No, I wouldn't actually, and that's one of my soapboxes. Is
Okay.
that all religious experience is not mystical?
Okay.
Yeah. Well, so I define mystical experiences as mystical unitive. There's no personality. There's no time or space. There's no content. There's a vast emptiness. There's no relationship. And contra that is what I call the interactive relational spiritual experience, which on the surface of it is a lot more typical of what my DMT volunteers report, which is a space full of content, and the personality is maintained, is verbal. There's interactions between the content of the state and the volunteer experiencing it. There's passage of time, the existence of space, even though they're altered. So that's kind of the opposite in a way. At least phenomenologically, from what I call the mystical unitive state. So the belief of a universal religious experience, I don't think is based in fact.
Okay.
The other question is, what is the relationship between any subjective experience and healing, either neurogenesis, neuroplasticity, improvement of depression.
Sure.
If you look at the literature every week, there's a new indication for psychedelics, or some field study indicating benefit. Mystical experiences, tobacco, alcohol, nature appreciation, openness, prisoner recidivism, flexibility, you name it. So I think there's a panacea-like effect which is occurring, and I think that is perhaps reflected in kind of a hyper activation of the innate healing mechanisms of the mind-brain complex. In other words, placebo. So right. I think what we may be seeing is that any kind of subjective effect, if it's intense enough, may activate neuroplasticity, neurogenesis, and that would be what we now understand to be the placebo response.
Right. And I don't know if he still speaks on this. Andrew Weil has certainly talked at great length about that's what we ought to be studying, is why there is this sort of intense placebo responsivity in some settings with some people, that there really is a remarkable righting of the ship, sort of ability the body has. One wishes we could harness. Do you, with any illness and its symptoms, there are people in medicine who sort of, want to be a bit nihilistic and sort of advocate that the symptom has adaptive value. The infected patient is well served by the fever. Don't treat the fever. It hastens healing. Thinking in that vein, can you think of any adaptive value that depressive symptomatology, the anhedonia, the neurovegetative features, the passivity, the withdrawal from life, does it serve any adaptive value? And are we usurping that or depriving patients of that by aggressively treating them for depression? I'm just curious.
Well, that's kind of out of my wheelhouse, as it were. But I do remember some data a long time ago, or speculation anyway, about depression stimulating more compassion, empathy, and things like that from the surrounding community.
Right.
So there may be some adaptive value. But still, I mean, you want to alleviate suffering. I mean, that's the role of the physician. So it could be it's a slippery slope. It's a fine line. All those ideas. I mean, you don't want to stand by while people are miserable.
Right. And certainly depression qualifies as that. They do suffer. You often don't hear about it because they're so withdrawn, but they are in exquisite suffering. And another question I wanted to ask while you're here, if you're willing to go into it, is what your thoughts are on Ibogaine. Will we ever get to a point where we embrace that? Will we see it invoked clinically for some of its vaunted properties? The allegation is out there that that opioid addiction can be cured by Ibogaine therapy. At least I've read that. Having been somebody who treated scads of opioid addicts as a hepatologist, trying to clean them up for other sorts of therapy, heaven knows we need therapies for that. Is Ibogaine a possibility?
Yeah, I do think it's a possibility. In both humans and in animals, it reduces craving, reduces withdrawal. So it is a good drug. It's kind of toxic. More so in women than men, and with cardiac underlying conditions. Back in the day, I helped the University of Miami get their Ibogaine study off the ground. I'm in the late 1980s, early 1990s. Deborah Mash in particular. So there were some concerns about its toxicity, and I was at the FDA meeting where they decided to not continue those studies in Florida to increase the dose anymore. There's a metabolite of Ibogaine which is called Noribogaine, which seems to have the same kind of anti-addictive effects in lower animals anyway. I'm unaware of its being put into humans, but I'm sure it's going on underground.
Right.
So that compound is less toxic and seems to be as effective in lower animals. David Olsen developed a non-psychedelic analog of Ibogaine, which he just published about 6 or 9 months ago.
Mm-hmm.
So that's a promising derivative of Ibogaine as well. And Atai is interested in Ibogaine. They purchased Dr. They I'm not sure if the word is purchased, but Deborah Mash's company is now under the umbrella of Atai. It's called Demorex.
Okay.
So they're actively looking at Ibogaine and its derivatives.
Yeah. Okay. That's helpful. I think one of the main toxic manifestations is refractory nausea, vomiting. Is that right?
Well, that's just an acute side effect, but the real
Okay.
toxicity, seems to be cardiac. There were some data, Mark Molliver at Hopkins, in the early 90s about, Purkinje cells in the cerebellum, but those were extremely high doses in primates, so, the human data. Well, you're steady on Ibogaine. You kind of, wobble around. And that could be related, to the cerebellar effects. But the cerebellar effects also, may be the path, to ending repetitive behaviors like addiction. So
Okay.
it's
Fair enough.
Complicated formula.
And I always like asking people like you who I think have taken profound journeys. Your life has been, it's not over. You're still on the journey. But I mean, tell us something that you've learned, a takeaway message about what you're about that you really don't think we get yet, that you don't think we're quite grasping, because bound to be something.
Well, I think it's the question of, what is a psychedelic, drug? Well, it's a mind manifesting drug.
Right.
And it only works on what's already in your mind, more or less conscious. So as time has gone on, I'm more interested in the content of the mind. Like as a psychiatrist anyway.
Right.
And I spent a lot of years studying Buddhism and practicing it. So it isn't as much. For most of us anyway, without serious illness, the mechanics of how psychedelics work, as much as what they're working on. So that's where I've kind of turned my focus the last 20 years or so.
Interesting. Okay. Jonathan, do we have questions? I
I've got a couple.
Okay.
One of them is from the aptly named Anonymous. Did anyone here attend the Psychedelic Medicine Conference in Miami earlier this week? If so, can you tell us about the general atmosphere there?
Did not attend. I think David Nutt, one of our consultants from the UK, attended, and I haven't had any feedback from him, but I know there was a major
Sure.
psychedelic conference going on.
Gotcha. Sam Tutu is regarding, I think one of the people you were talking about, that she had written a book on self-microdosing of LSD and the struggles of depression and suicide. I forget the person in question, but I think that was Dr. Strassman. You had just been talking about her a few minutes ago.
Yeah, I think the book is called A Very Good Day, and the author is Waldman. I yell at Waldman. Yeah. So she took microdoses of LSD to help with her depression, and it was quite helpful. Well, when you're talking about microdosing, I think it's important to think about three levels of microdosing. There's completely so there's completely inactive doses, where you don't feel anything at all. And if you take those daily, I think the same kinds of receptor changes that occur with the SSRIs take place. And you have an antidepressant response. If you take a slightly higher dose, it's a bit sort of stimulatory, but it's not psychedelic. And I think if you took those daily as well, there would be the same SSRI, like responses. And you take, perhaps, slightly higher doses, it gets a little sparkly, and you have an indication of what's in store if you increase the dose even more. So I think it's important, to think about, what kind of microdoses you're talking about. The microdose effects also, may reflect that the that the neurotox or that the psychoplastogenic effects, don't necessarily require psychedelic effects.
Well, Chris, I've got a question, and I hope I kind of parse this out correctly. we kind of see, at least on the DMT side, what your expectations are, right? We see where we are now. You're hoping 8-10 months, something like that. Again, putting a number out there that may or may not be quite correct, to be in phase 2. What's the kind of pathway for Algernon to get there? If we see kind of where we are, where we want to be. What's the pathway to get there?
Well, for a company like Algernon, there are really two elements to moving into a phase 2.
Yeah.
They're both big hurdles. One is the regulatory approval.
Yeah.
So that, and it may sound obvious, but people should realize that, of course, you have to have
It's a gauntlet.
You have to have good phase 1 data. You also, by the way, need to be working. Your data needs to interest investigators. Okay, because you've got to convince PIs that your data, animal data, your hypothesis, the safety is interesting enough to them that they want to encourage their patients. So I'm not concerned about that for a DMT. I think we've had lots of interest. It's a much needed area. Then the second hurdle is simply having the capital to do it.
Yeah.
And how big does that study need to be so that you can get statistical significance, good p-values, or is it a smaller 2A, phase 2A, where you're looking for signal and then you're going to go into a bigger phase 2B and then hopefully move it into a phase 3. So really regulatory, and second is having access to capital to run a phase 2 trial. And that could be, typically speaking, you're looking at somewhere in the $2 to $5 million range to run a phase 2 study. It's quite wide because it depends on how large the study is, what the incident rate is, it chronic or acute. If you have to study a patient for 1 year, like IPF, your costs are going up. Sometimes you have to cover the cost of standard of care. Standard of care for IPF is about 80 to 90,000 US per patient. So, but that's really the hurdle is regulatory and capital. So I hope that answers that, Jonathan.
Yeah, mostly. Obviously, I've got questions about the jumping of the hurdles, but I'm not sure if you're prepared to go there.
Look, we have what I can say, and I mentioned to Doc earlier, we have a plan to uplist to the NASDAQ. It is a process. Algernon is in a very good capital position right now. We received about $2 million from our research work in Australia. That was a refundable credit, not just a tax credit. So, we reported about $3.5 million in the bank. Why, being asked, why do we want to go onto the NASDAQ? And I believe we need to be continue to put the company in front of investors that can appreciate and have an understanding of the work that Algernon's doing so that we can improve our valuation. I think it is clear in the last 4 to 5 months that, in fact, a majority of investors were in the stock because of COVID. And when we announced we weren't moving Ifenprotal forward, there was really sort of a onto something else. And so, a company like Algernon will go back to the markets from time to time to access capital to continue to drive research. That's all about timing.
Sure. I think you had mentioned, and if I need to stop, let me know, something with regard to Joe Rogan. Obviously, not your intention here, especially considering, you could become sort of almost meme-esque, if you will, should he kind of get some interest in the stock, which I find both fascinating and frustrating when we have someone like Dr. Strassman, who's eminently knowledgeable and capable, and yet what may draw more interest is the incredibly brilliant KOL or the guy who's incredibly popular. It's a very strange dichotomy in this world, but, I'm sure you guys are aware of the potential power of that avenue.
Yeah. I mean, there's just so, if you think about how many companies, how many compounds, how many sectors, how many industries that are publicly traded that are relying on interest from investors, you have IR, you have PR, you want people to learn about your company. Everyone has limited time. So Sometimes it's you have to do whatever you can to get that message out. Sometimes it's better to be more conservative and be a little more thoughtful. Sometimes it's not about quantity, it's about quality. I'm afraid there's no one answer. It's an aggregation of a number of different strategies. That's what I found. And
Totally fine.
somebody says they've got the magic sticks and they know how it all works. Run like hell.
I like it.
Solid advice. Another question from this one from Peter Hoffman. Has Algernon dropped his other potential products to focus on DMT solely? If not, what is the other status? What are the statuses for the other drugs?
Definitely haven't dropped. We are currently running an IPF and chronic cough trial in Australia and New Zealand, brutally affected by COVID. They literally having shut that country down for months and months and months. We've added a new site. We're hoping to complete the full enrollment of 20 patients in that study. We've applied based on positive trending data from the cough part of that study with the US FDA, with a pre-IND for our own cough study in the US. We also acquired some technology from Dartmouth College, brought on Dr. William North, and we announced a program for ifenprotal to explore small cell lung cancer and pancreatic cancer. Those cancers seem to express the NMDA receptor, and blockade, or rather antagonism by ifenprotal seems to have an effect, a positive effect on reducing tumor growth and so on. So ifenprotal is our key program along with DMT. I have one or two other compounds that we haven't talked about yet. And so one of them is another drug that comes from the Far East. So that's all I'll say on it. But we have other batters, or pitchers rather, in the bullpen, to use a baseball analogy.
Very interesting. Sam22 is asking a question, maybe for you, Dr. Strassman. Would it make a difference in the amount of dosing used in stroke victims depending on the stage of the stroke he or she is in?
I'm not quite qualified to answer that.
Well,
but if you're concerned, if you're concerned about increasing the size of a hemorrhagic stroke, I think the fact that low doses don't raise blood pressure would militate against those concerns. I think the animal data would be more enlightening regarding what kinds of doses are required at what stage, acute or recovery. What do you think, Chris?
I think that I don't disagree with you. I think that we're only going to learn so much about the duration of treatment and the benefit and timing from animal studies. And I think since we're safe, we have to conclude our phase 1, but historically it's safe. I think that we're going to be far more informed by starting into a phase 2 cautiously, where we're going to pick our spots. What's the best time to treat post-injury? After all, it's rare for you to really know when the injury occurred. You might be found on your couch unconscious. You may wake up in the morning and see your spouse isn't responding. If you're standing with a partner and you're talking and you're getting confused, it could be happening right then. So you've got that better chance for them to know when the injury is. Mm-hmm. But for the most part, if we can't really tell when that injury is, what does the sum of the timing show about when we should start to treat, and then the duration? I think based on the literature, Rick, and maybe there seems to be about a 2-week period where there's response from the neurons in terms of this growth or neurogenesis or neuroplasticity. Is that a phase? I'm not sure. But how much to give the patient? As I said earlier, we seem to see 2 parts of it. There's this initiation phase where it's almost like it's getting the machine going, and then there's activity is increasing in sort of the 2nd, 3rd, and 4th hour compared to the 1st hour. So we have lots to learn about it, but what's important is I think we can do this safely. We can explore in our phase 2. We don't want to totally shoot in the dark, but we'll be better informed by actually running the phase 2 studies, I think.
Agreed.
With respect to the phase 2 studies, will those be single boluses or continuous infusion?
Our plan is continuous infusion.
Oh, great. Yeah, because there are a couple of groups that are working on that. There's the Imperial College group, Chris Timmerman and his colleagues, and there's a group in Switzerland that's working on a continuous infusion model as well. So yeah, that's good to hear.
This is another one from Anonymous. This one's for you, Dr. Strassman. Do you have any thoughts on the possibility of non-hallucinogenic drugs which don't have the problem of FDA scheduling, such as 2-bromo LSD for depression or alcoholism, PTSD?
Yeah, well, yeah, the whole issue of the non-psychedelic psychedelics is going to be a huge upcoming area of interest. These compounds seem to demonstrate this same kinds of antidepressant and of anti-addictive efficacy in animal models as the classical psychedelics do. But in animal models, they don't induce the head twitch response, which is one of the gold standard indicators of your psychedelic subjective effects. David Olsen's been working on these kinds of compounds. Brian Roth at UNC is developing those kinds of compounds. The two main ones, which are coming out of UC Davis and Dave Olsen's lab, are that one of them is an Ibogaine derivative, and the other is a DMT derivative. So BOL is one. There's going to be a slew of these so-called non-psychedelic psychedelics. They bind to the 2A site. They cause the same conformational changes in the receptor. The same cascade of downstream effects. But what is their effect in humans? There's a little bit of BOL data. The bromo LSD compound. But with respect to these other compounds, we don't really know anything about what they do in the human yet. Yeah, and strictly speaking, if you could kind of activate an antidepressant response, comparable to psilocybin without the psilocybin subjective effects, you'd be that further along, because a lot of people don't want to trip. All they want is their depression to be improved.
Rick happened to be an advisor for a company working with that compound, and one of the great virtues for them is their ability to CMC, CGMP drug is makeable without ever coming near LSD. You can kind of backwards assemble it and bypass a lot of regulatory hurdle and hoop by never having to go near LSD to make 2-bromo LSD. They can sort of piecemeal assemble it in a reverse way. So that makes life way easier for them.
Well, there's quite a few new compounds in the pipeline. I've been consulting for the Alexander Shulgin Research Institute, and they're sitting on a treasure trove of some of Sasha Shulgin's compounds. Some of them are in the public domain because he published them in his books. Others are just sitting on the laboratory shelves waiting to be looked at.
Another question is from Karen regarding the COVID trials. Is there any route to try ibuprofen perhaps earlier in treatment, maybe right after COVID diagnosis, and kind of see, if maybe there'd be a more robust effect?
So it's for us, it's really one of resources.
Yeah.
And we looked at, when we started this last March, everybody had just been locked down.
Yeah.
February, March. Then we found out about H5N1. Then we decided to throw our hat in the ring. And we decided to go after the very sickest of the sick people, and that's folks who had been, because there was no talk of vaccines last March.
Right.
So it wasn't even on the table, and people would have looked at you incredulously if you said, well, we're going to have a vaccine by December.
I mean, last March seems like 3 years ago to me. It really does.
And keep in mind that 85% of hospitalized patients get better on their own.
Right.
You're up against a pretty tough hombre. We went in and we saw that the data really wasn't strong enough to suggest moving it forward to a phase 3. Would we want to go back for another round? Rocky 6 or 7. I'm not sure where. I just saw something about Sylvester Stallone this morning.
We're in double digits now. But yeah. Obviously the whole COVID landscape has changed, especially now with the 2 oral options from Merck and Pfizer. So I get where you're going with that. So that's
I think what we were hoping for ifenprotal was for the Merck drug.
Yeah, pretty much.
Where this had, but so we've been looking. We're looking at there are challenge studies now. There's a challenge study in the UK where people have agreed to be infected, and ethically it was a big thing to be infected with COVID, because if you're going to do a post-infection prophylactic, you're talking about 30,000 people. So you're talking about a scale that we didn't have the support. We don't now. And, do we want to get back into that? I do think that ifenprotal has demonstrated some antiviral qualities in the preclinical data.
Yeah.
There may be something worth to looking at its antiviral. We've been discussing it at our scientific and medical advisory board level.
Yeah.
I don't think right now, with the way everything is going with the vaccines and the antivirals, that we saw enough to want to try to throw our head back in the ring.
Jump back into the pool.
Be hard to do right now.
Well, switching gears, Chris, this one from Norman Glassman. Are you concerned about the possible relationship between current share price and your ability to raise capital, let alone secure NASDAQ listing?
I'm not. And I think that right now, if you look back at March, we were at 5 or 6 cents. Then we announced the COVID trial. We got up to 50, 60 cents. We've traded, investors may be interesting to hear this, 1 billion shares have traded in Algernon. When you aggregate the CSC, the OTCQB, I think it's at a tremendous entry price right now. I don't think we'll have trouble accessing capital. I think the repurposing story just needs a bigger audience. I believe we should stay with that repurposing story. I think that we have a very capital efficient model. We're going to work on getting that message out to new shareholders, and uplisting in the US is one of the key initiatives that I really strongly believe, obviously, that we need to do.
Okay.
I will commend Chris I've never seen anybody make a dollar go as far at a corporate level as you do, in terms of what gets accomplished investigation wise for very limited funds. And it's just a question of whether it's going to beat up the circumstances to make it a NASDAQ uplistable entity.
Well, the best is yet to come.
Yeah, I agree.
We have no further questions, right? Oh, Dr. Strassman.
Yeah, well, so Chris, do you consider the SPAC route?
We've looked at it, but there are some elements that are less appropriate for an already publicly traded entity compared to we're already on the OTC QB, we're on the CSE. So it was part of our review process. There have been a couple of companies, Doc knows one of them, Diamedica. They're running a phase 2 stroke trial in China. I think they're $130 million US market cap. So think a bit about that phase 2 trial for stroke on the NASDAQ. Zortex just came onto the NASDAQ, another Canadian company. I think we have a multi-dimensional aspect to what we're doing that would also make it very interesting and appealing and attractive to investors. But so I do think it's, there are some benefits to starting out on the CSE because of either RTO or CPC, but depending on the sector, it's not for everybody. I do firmly think that a clinical stage pharma drug development company is going to be better off on with a US listing.
I agree.
Jonathan, I know you need to take off. We'll see. Appreciate your help.
Thanks,
Yeah.
Doc. And
Yeah,
great
thanks.
to see you. And Dr. Strassman, great to meet you. Really appreciate your knowledge. And
Well, thanks, Jonathan. Take care.
see you, Chris.
See you, John.
I think that's it for questions at the moment now. But yeah, Chris, we're behind you. I think that with the share price being at 5%, it's at 5 cents. It's easy to become emotive and want to lob tomatoes. And we try to counsel people that's not an adaptive response to this at all, and that the company does have things going for it. They're not all things we can discuss right openly just now. And you have to have faith that Chris is not leading us blindly over a cliff. He's not. He's not. So, and I think I'll just leave it at that. Guys, I would suggest we bring this plane in for landing. We've been at it going on an hour and a half or so. Dr. Strassman, this is a wonderful session. Really would love to have you back again and just get you going again and really hear what you have to say as you've done today. It's really I love sessions like this. We've had a couple of very interesting psychiatric domain guests following Chris as we have. So they make for great conversation. Chris Bryan, for those we haven't really unveiled CB as we call him yet, but wonderful addition to the Algernon team. Glad you're there, Chris, and you're you're a remarkable person. You're quite the analyst, and your presence is valuable to all of us. So
and he's got the beard. He's got the beard going, which is kind of it's a playoff thing without the playoffs. But
nice. Well, folks, if there's no other questions, really a great session today. Sorry for the weather. Some of you all are up north there, but I think you all are better at handling it than we are down where I am. So I'm virtually Tierra del Fuego, kind of south of the Mesa-Dixon line. So anyway, why don't we call it a wrap for today? Dr. Strassman, again, appreciate your presence and hope to have you back whenever you want to come back. Just we'd love to have you. And
sure, just let me know. Yeah, send me an email. Let me know when you want to chat, and I'd be happy to.
It'd be great. And we'll have a videotape of the event up to the site probably this evening, and I'll personally send links to all of you of that as well. So,
hey, Doc.
Folks, sorry, Chris, go ahead.
No, I just wanted to say thanks again. Good seeing you, and appreciate having a chance to speak to the BioPub group.
Yeah, always welcome, Chris. It's great to hear from you. Great to get an update. It's sort of we kind of lopsided in our coverage. We covered so heavily last year and kind of not so much this year, but you've had other fish to fry, and that's okay. So we'll maybe try to even out a bit in the 2022. So anyway, folks, we'll see you online at the BioPub. Thanks for being here, and let us know if you have questions, comments, or need help. If you have anything you'd like to ask that I can forward to these gentlemen, be happy to do that. And really a great session. Have a fine weekend, everyone. And I'll be in San Diego next week for a conference. We'll have our final webcast of the season next Friday. It's on a non-biotech play, as you know. And then happy holiday season to everyone. I guess it's sort of impending, not too early to call it holiday season. So anyway, thanks, everybody. Have a great weekend. See you soon. See you online.
Thanks.
Thanks.